कॅम्पस मध्ये रेस्टॉरंट असतं.
सर स्ट्रीमिंगच्या
युनिव्हर्सिटीच्या बाजूला बाहेर जायचं, 5 मिनिटं चालायचं आणि त्या रेस्टॉरंट मध्ये मधल्या वेळामध्ये अर्ध्या तासामध्ये 20 मिनिटांमध्ये खाऊ नये.
काही आपल्याकडे प्रोसिजर हे पाहुण्यांचा सगळं आदरातिथ्य यामध्ये
फार प्रोसिजर आहे. उपचारामध्ये आपला वेळ फार जातो.
पुन्हा एखाद्याचा हार राहिला चुकून द्यायचा किंवा फूड राहिला तर फार मोठा इश्यू होता.
प्रचंड राग येतो परत.
आपल्याकडे राफेल आले तरी त्यांची पूजा केली गेली.
सर अनम्यूट करा. सर अनम्यूट.
सर चौधरी सर. हॅलो चौधरी सर.
हा बोला.
हा आपल्याकडे ती राफेल आली तरी त्यांची पहिल्यांदा पूजा केली गेली ना.
हो हो बरोबर.
आपल्याकडे सुरुवातीला ते वंदन करण्याचं आहेच.
हो ते आहे, बरोबर आहे. आपल्या परंपरा बरोबर आहेत, पण आपला वेळ मग त्यात फार जातो.
हा नाही ते आता आपण जितकं आटोपशीर करता येईल तेवढं
आटोपशीर करण्याचा प्रयत्न करायला पाहिजे.
अनिरुद्ध जॉईन झाले. अनिरुद्ध हे जॉईन.
अनिरुद्ध झाले.
जॉईन झाले.
हा थोडे पार्टिसिपंट्स येत आहेत अजून.
हा ठीक आहे. आणि आणखीन वैशिष्ट्य असं की शार्प डॉट त्या टाइमला सुरू होतं त्यांचं. आणि आता तुम्ही सर्टिफिकेट सर पार्टिसिपेशन ज्यांना आहे त्यांना ई सर्टिफिकेट सध्या देता. तर अमेरिकेमध्ये कॉन्फरन्स अटेंड केल्याबद्दल अटेंडन्स सर्टिफिकेट देतच नाही. नोबडी आस्क्ड also. I was the first person to ask for attendance certificate. मला म्हटले का कसलं अटेंडन्स सर्टिफिकेट? म्हटले आम्ही काही देत नाही कॉन्फरन्सला. अहो म्हटलं मी कॉलेजमधून आलोय, इथे युनिव्हर्सिटी कॉलेज रिप्रेझेंट करते आणि मला अटेंडन्स सर्टिफिकेट लागेल, नाहीतर मला म्हणतील कुठं फिरायला गेला होता काय कॉन्फरन्सला.
आपल्याकडे ते अटेंडन्स सर्टिफिकेट महत्वाचं आहे. अटेंड नाही तरी चालतं.
मग म्हटले कॉन्फरन्स संपल्यानंतर काय कुठल्या फॉर्मॅट पाहिजे तुम्ही द्या. आणि मग युनिव्हर्सिटीच्या लेटर हेडवर त्यांनी कॉन्फरन्स लेटर टाईप केलं आणि तिथल्या डीननं साईन केलं ते आणि माझ्या स्वाधीन केलं. म्हटले तुम्ही एकमेव आहात की अटेंडन्स सर्टिफिकेट मागताय. जगातले सगळे लोक आलेले आहेत. म्हणजे विश्वासाचं वातावरण आपल्याकडे कमी असल्यामुळं तिकडे काय असते, त्या बाहेरच्या युनिव्हर्सिटीमध्ये लोक विश्वास ठेवतात. हे गेलेत कॉन्फरन्सला म्हणजे गेले, आपले प्रोफेसर कॉन्फरन्सला गेले, मी कॉन्फरन्सला गेले, ते अटेंड करणार. आपल्याकडे अविश्वास दाखवतात. आपल्याकडे गेलेत म्हणजे काय, फिरतायत का कॉन्फरन्स अटेंड करतायत. तर थोडा विश्वासाचा भाग आहे आपल्याकडे थोडा कमी आहे. 2 अनुभव मला खूप लक्षात राहण्यासारखे आहेत.
होईल तसं. ऍडमिट कर लोकांना. हो. Our meeting is being started within few minutes.
Join.
69.
सर तुमचा लाईट थोडासा असा उलटा पडतो. आवाज नाही सर तुमचा.
Yes, sir. माझा? हो.
Aniruddha?
Yes, sir.
Do you hear me?
Yeah, we are absolutely.
Okay. Shall we start?
Sure.
Chaudhary sir, shall we start?
Yes, sure.
भालेराव सर.
ऍडमिट करायचं बंद करतो. तू फक्त तेवढंच काम कर.
अरे किरण
भालेराव सर जॉईन.
भालेराव सर सुरू करायचं.
हो 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 सर आहेत ना.
आहेत.
मलाच घुमतो आवाज.
मग तो सगळ्यांना दिसते मला.
बरे ब्लॅक
सगळ्यांनी ऍडमिट करायचं. So the countdown begins. 5, 4, 3, 2, 1. I am Ravindra Bakre, convener of International Webinar on Biomedical Science and Bioinformatics. Good evening, everybody, and I wish you happy Teachers' Day. Honorable resource person, Mr. Aniruddha Pandit, bioinformatician and web developer from University of Glasgow, UK. Honorable Dr. J.S. Chaudhary, Secretary, Janta Shikshan Sanstha Y. Honorable Dr. E.B. Bhalerao, in charge Principal of Kisanir Mahavidyalaya Y. Professor Dr. S.R. Sawan, Member of Academic Council, Shivaji University, Kolhapur. Dr. H.D. Kanse, Organizing Secretary of this international webinar. Mr. V.B. Karde, Vice Principal of Commerce. Dr. B.B. Agedkar, Vice Principal of Arts. Dr. S.P. Kamre, Coordinator of Internal Quality Assurance Cell. Ladies and gentlemen, good evening. On behalf of Janta Shikshan Sanstha's Kisanir Mahavidyalaya Y, Department of Zoology and Internal Quality Assurance Cell, I take this opportunity to extend a very warm welcome to all of you in this one-day international webinar on biomedical science and bioinformatics, organized through Zoom cloud meeting and live YouTube streaming today. All of you know COVID pandemic has been causing a lot of havoc to human endeavors all over the world. All educational initiatives closed for the students. Classroom teaching has been closed for students. Class, Classroom teaching, organization of curricular and extracurricular activities at the campus is no longer possible. Nevertheless, thanks to the internet and online apps, it has become possible to conduct online classes as well as online organization of webinars. Ours is one such attempt to organize this international webinar on biomedical science and bioinformatics. Historically, the term bioinformatics did not mean what it means today. Paulian Hogeweg and Ben Hesper coined it in 1970 to refer to the study of information processes in biotic systems. A major activity in bioinformatics is to develop software tools to generate useful biological knowledge. Bioinformatics has become an important part of many areas of biology. In structural biology, it aids in the simulation and modeling of DNA, RNA, and protein structures as well as molecular interactions. Most bioinformatics specialists are employed by pharmaceutical companies that use biological and genomic data to develop new drugs. Others work for biotechnology companies that develop new medical treatments and products. Some work for government health agencies and hospitals. Omic technologies are primarily aimed at the universal detection of genes, that is genomics, mRNA, that is transcriptomics, proteins, that is proteomics, and metabolites or metabolomics in a specific biological sample. Mass spectroscopy is the most common method used for the detection of analytic in the proteomic and metabolomic research. Multiomics or integrative omics is a biological analysis approach in which the data sets are multiple omics, such as the genome, proteome, transcriptome, epigenome, metabolome, and microbiome. In other words, it is the use of bioinformatics. Bioinformaticians apply information technology to biological, medical, and health research. They use computational tools to gather and analyze data in fields such as population biology, genetics, and pharmaceutical development. Today's resource person, Mr. Aniruddha Pandit, is a young scientist, native of Y, is working in bioinformatics, and we had been always in touch with each other by social media. It was my feeling that he should talk to my students and the researchers in this field to make them information available about his work and current scenario in this field. He instantly agreed to join on Zoom meeting for all of us, and now he is with us. I cordially thank him for accepting our invitation to talk on this recent biological field. I hope that this webinar will make our purpose fruitful. I am thankful to the resource persons and participants for their registration, cooperation, and joining with this evening August gathering for this webinar. Now we are proceeding towards the inauguration function. I request Dr. Sunil Sawant to take the charge.
Dr. J. Chowdhury, today's chief guest for this inaugural program of the international webinar on biomedical science and bioinformatics, is the secretary of our Janta Shikshan Sanstha Y. Dr. Chowdhury enjoys the rare privilege of being associated with our institution right from the beginning. He had his undergraduate and postgraduate education in the erstwhile Arts and Commerce College of Y. He also received a degree in law. Immediately after completing his post-graduation, he joined as a lecturer in our college. Later, he became the head of the Department of Commerce and Management. He had a rare opportunity to work as a principal of our technical computer lab for many years. Even after his retirement, he was closely associated with our institution as an active member of our alumni association. Recently, he once again joined our fraternity, this time as a secretary of our district Janta Shiksha Samstha. He has worked as a research guide. 12 students have been awarded PhD and 16 MPhil under his guidance. He has published more than 12 books on commerce and management. He has also been closely associated with Rotary International and contributed to Rotary in different capacities. Above all, he is a born teacher and likes to interact with students and teachers whenever he gets an opportunity to do so. One of the resource persons of today's webinar, Dr. Abhijit Chowdhury, is his son. The most remarkable thing about his family is that all the 4 members of his family, he and his wife, Abhijit and his wife, are PhD holders, and all of them enjoy the pursuit of knowledge all the time. I invite Honorable Dr. J.H. Chowdhury to make his inaugural address in this international webinar on biomedical science and bioinformatics.
Thank you, Dr. Sawant, for a very nice introduction on this very important occasion. So friends, we are meeting today for online webinar, Zoom webinar, on a very important theme on biomedical science and bioinformatics. We have good resource persons, because normally when we have international conference, we say that we have international conferences, we have national when we have national conferences. Most of the time we see that only teachers, students from Maharashtra are joining, but we call it international sometimes. Sometimes, sorry, national we call it, or sometimes we call international, but hardly there is any international participation. But this time we are lucky to have 2 resource persons from two countries. Aniruddha Pandit is from England. He would be a resource person. And Dr. Abhijit Chowdhury will be also another resource person who is a professor in California University. So we are glad that in true sense of the term, we are having an international seminar or webinar this evening. So, in-charge principal, Dr. Bhalerao, who is the chairperson of the inaugural session. The convener of the webinar, Professor Bakre, Secretary Dr. Kanse, IQAC coordinator, Mr. Kamble, Dr. Sawant, the vice principals of the college, all the participants, teachers, professors, students, researchers, and friends. It gives me pleasure in inaugurating today's international webinar. As Professor Bakre said, sir, bioinformatics is a very important science these days, and there are many career opportunities to our young scientists and researchers. Bioinformatics, as you know, is the science and practice of utilizing computer technology to handle the data used to address complex biomedical problems. And so the use of computers in handling biomedical problems is becoming a very important and novel field for those who want to pursue their further career. As far as the role of zoology is concerned, we know that we need to understand the basic concepts in the zoology science, especially understanding basic concepts such as evolution to predict changes in species of the future, to understand our environment, prediction of climate change patterns via observing changes in species, understanding changes in genetics because of environmental factors. Then another facet of this subject is animal models for studying cancer and other diseases, which are the major problem these days. One aspect of this is research on ways that diseases are spread and progress. Another aspect is developing and testing treatments and drugs to cure these diseases. Then one more aspect of the study of zoology is pet animals as models for human exposure to factors like smoking, toxic water, pesticides, poisoning, etc. Then the next aspect of this very important subject is many career opportunities. As Bakre said, sir, that there are several career opportunities coming forth for our young graduates and researchers. So some of the career opportunities in this field would be lab-based research. Second is computer programs and statisticians, computer programmers and statisticians, technical writers, medical doctors, veterinarians and animal care personnel, design and maintain medical devices, research equipments, animal housing and laboratory facilities. So these are several opportunities for our young graduates pursuing these subjects, zoology subject. And through this webinar, I hope that our students, our teachers would get a new insight for developing their capabilities, developing their knowledge, their skill in the field of zoology. So, friends, with these words, I again congratulate Professor Bakre, principal of the college, and his colleagues for organizing this event, this international event this evening. On behalf of Janata Shikshan Sanstha, its president, Sri Pratapra Bhosle, Vice President, Sri Gadve sir, and all the office bearers, I extend my warm greetings to all the participants in this seminar, in this webinar, and hope that today's seminar would be a great success. Thank you very much, and I finally declare that the webinar is inaugurated. Thank you very much.
Now I request Dr. Sunil Sawant to introduce today's chairperson, Dr. E.B. Bhalera, in charge principal of Kisanir Mahavidyalaya Vai.
Dr. E.B. Bhalera, the principal of our college and the chairperson of this inaugural program, has been teaching in the Department of Botany of our college for more than 30 years. He has his PhD from the renowned University of Pune. He has won the prestigious NSS Program Officer Award of our Shivaji University, Kolhapur. Dr. Bhalerao is a very sincere and passionate teacher of botany. He has played a very important role in the development of mango orchard on our campus, just in front of our Jai Kisan Boys Hostel. His son Ganesh is agricultural officer in Bank of Maharashtra, and his daughter Sonali has been pursuing her doctoral research in medical biotechnology at the most eminent center of science research located at Chandigarh and affiliated to JNU New Delhi. I make a humble request to Dr. E.B. Bhalerao to talk about our college and give his chairman's address.
Thank you, Professor Sawant sir. Ladies and gentlemen, on behalf of Janta Shikshan Sanstha Y and Kishanur Mahavidyalaya Y, it gives me great pleasure to welcome you all in the international webinar on biomedical science and bioinformatics, organized by the Department of Zoology and IQAC of our college. Our college is one of the best rural college affiliated to Shivaji University, Kolhapur. It was established by Sri Kishan Veer, a veteran freedom fighter, and by Padma Vibhushan Tarkatirtha Lakshman Shastri Joshi, a renowned Sanskrit scholar. The college made a humble beginning with only arts and commerce faculties in 1962. It later added MCom, MA, Science faculty, and recently in 2012, the management has started MSc Organic Chemistry as well. Many eminent leaders and scholars such as Yashwantrao Chavan, founder Deputy Prime Minister of India and former Chief Minister of Maharashtra, then Appasaheb Pawar, former Vice Chancellor of Shivaji University, Kolhapur, Dr. Jayant Naralikar, world-renowned scientist, and several others have paid visits to our college from time to time and made valuable guidance to our students. The management of Janta Shikshan Sanstha is knowledgeable and service-oriented. It has made remarkable contribution to the changes in infrastructure of our college, taking into consideration the changing needs of students' population. With the help of grants from the UGC, the management constructed women hostel and indoor sports hall. Our management has shown a great sense of social commitment by undertaking a noble project of constructing boys hostel for the students belonging to families of the farmers who have committed suicides. This project has been funded by the principal, the teaching and non-teaching staff, and the alumni of the college. We all feel proud of our management. In 1962, the strength of the college was only 80, but at present, it is more than 6,000. The growth of the college is not only quantitative, but also highly qualitative. Our students have excelled in the academic, cultural, sports, and NCC fields at the university, state, national, and sometimes international levels. Many of our students have occupied good positions in administrative in administration, industry, banking, research, and corporate work. The faculty of our college is well qualified. A good number of teachers has obtained MPhil and PhD degrees. Some of our teachers have successfully completed major and minor research projects. Our administrative staff is active and hardworking. The reason all participants have joined this Zoom meeting is because the Department of Zoology and ICAC of our college have organized this international webinar, and the topic of the webinar is extremely relevant. I hope all of you will enjoy the deliberations. Once again, I welcome you all here. Thank you.
Thank you, Dr. A.B. Bhalera. Now I request our IQAC coordinator, Mr. S.P. Kamble, to offer vote of thanks.
Thank you, sir. Honorable chief guests, respected resource persons, and all the participants. It's my privilege to have been asked to propose a vote of thanks on this occasion. On behalf of Janata Shikshan Samstha's Kishanveer Mahavidyalaya Vai, Department of Zoology and IQAC, I extend my sincere thanks to Honorable Pratap Rao Bhosle, alias Bhau, the President of Janata Shikshan Samstha, and all the directors for their constant support and motivation for the organization of such memorable events. I would like to extend a special thanks to Honorable Secretary Dr. J. Chaudhary sir for his presence and for sparing his valuable time out of his busy schedule. We are really enlightened with your knowledge and presence. I must mention our deep sense of gratitude and appreciation to the today's resource persons, Dr. Aniruddha Pandit sir and Dr. Abhijit Chaudhary, for accepting our invitation and for their presence in this program. I also wish to express my gratitude to our Principal Dr. E.B. Bhalera sir for providing encouragement and support. A special thanks to Dr. S.R. Sawant sir, Head Department of English and member of Academic Council, Shivaji University. Dr. Kent Sontake sir, Dr. Vaidya sir, Dr. Kanse sir, Lieutenant Samir Pawar, Dr. Agadkar sir, Vice Principal of Art Faculty, and Mr. Karde sir, Vice Principal of Commerce Faculty, and the convener of this webinar, Mr. Ravi Bakre sir, all the organizing committee members, teaching and non-teaching staff of our college for their unflinching support and coordination. I am very much thankful to all the participants and students for active participation in this international webinar. At last, I express thanks one and all with these warm words and a kind message. I move toward the end of the inauguration function, and I hand over the mic to the convener of this webinar, Mr. Bakre sir. Thank you, sir.
Thank you, Mr. S.P. Kamble. With this, we have concluded the inaugural session, and now we are moving towards session first, and I hand over the charge to Professor Dr. Sunil Sawant for introduction of today's speaker, Mr. Aniruddha Pandit from University of Glasgow.
Mr. Aniruddha Pandit, bioinformatician and web developer at University of Glasgow, United Kingdom, is a highly motivated, organized, and adaptable research scholar with a remarkable mastery on bioinformatics and biology. After completing his MSc in zoology from Shivaji University, Kolhapur in 2001, he undertook PG diploma in bioinformatics at National Institute of Bioinformatics in 2002. Thereafter, he completed his Master of Science in Bioinformatics at University of Aberystwyth, Dundee, United Kingdom in 2004. As a renowned bioinformatician, he has a very extensive experience in both commercial and academic research environments. He has exemplified capabilities in design and development of applications, workflows, and data analysis pipelines for various data types, including next-gen sequencing data. He possesses excellent project management skills, and he has led small groups and projects successfully. He enjoys undertaking new and challenging problems and likes to resolve them in a rational and logical manner. He aims at undertaking more challenging roles and responsibilities and making a substantial contribution to academia and industry as part of his career progression. Many of his research papers have been published in the prestigious journals at the international level. Mr. Pandit is a lead bioinformatician for various projects, including N-Euro StressPEC, an international 14-member consortium project spanning both research and industry, and the international collaboration between UK and Brazil, BBSSRC funded project, SCPTRE, and FAP ESP BBSRC funded project on insecticide resistance management. He is also a member of the team for projects such as Fly Atlas and Fly Atlas 2. He is also responsible for data analysis of transcriptomic, genomic, and metabolomics data, website and database web application development, pipeline creation, and software module creation. He has supervised undergraduate honors and master's students' final year projects as part of their degree curriculum. He is an expert in successfully undertaking many duties and responsibilities. Some of them are identification of objectives, planning, developing, and implementing projects in a timely fashion, design, development, maintenance of project websites and database web applications, database management, data mining, and data curation of relevant data, creation of pipelines, drafting publications, documenting research output in the form of project reports and presentation of his work at conferences and consortium meetings, reviewing costs, specifications, and purchase of servers, computers, and other peripherals, and their maintenance within the laboratory. Collaborating with other laboratory members with their research by performing necessary bioinformatics analysis as per their requirements. Dissemination, brand management, data management plan creation, and social media engagement. Mr. Aniruddha Pandit was born and brought up in Y. His father, Dr. Ashutosh Pandit, was the founder, member, and director of the then Y Akshar Institute, the institution which did the pioneering work for the schooling of the mentally retarded children. His father did his doctoral research on the training of the parents of the mentally retarded children of Vai Taluka and submitted his thesis to MS University, Baroda, Gujarat. His father is widely known for the development of Akshar Samayojit Vartan Pariman, a scale to determine the strength and the needs of the mentally retarded children. Mr. Aniruddha, the son of such an illustrious father, has been making his own remarkable contribution to the field of bioinformatics. I invite Dr. Aniruddha Pandit to make his presentation on his topic.
Thank you, Dr. Sunil Sawant, and welcome again, Dr. Aniruddha Pandit.
Thank you, sir, for that. That is very kind of you to say all these kind words. I don't know if I'm that much, as much as you've said. I will try and give a good presentation today on the topic. I will just share my screen. And, hello to everybody who who I can't see right now, but are here for the talk. I'm just going to start my presentation now. Is that visible to everyone?
Yeah. Yes.
Visible, no problem.
Excellent. So again, thank you for giving me the opportunity to talk here. This is a great experience for me just to be here. As a Viker, I think I'm very honored and privileged to be invited to talk here. So yeah, I'm just gonna try and get through my presentation as quickly as possible because I know time is of the essence here. But I wanted to just say namaskar. I am from Y. I've always been a Yiker at heart. I've been in Glasgow and Scotland now for almost 17, 18 years. The two pictures you see side by side are of Y in Glasgow, as you can see. There's not much difference. There's a river running through them, and there are bridges there, and it's like home, except I'm not in Y. Right. So coming back to my talk, as Dr. Sound has already introduced me and my career, I'll just do a quick overview. I did my 10th standard in New Era High School in Panchkani. I did my 12th in Kishanvir Mahavidyalaya back in 1996. I passed out my 12th standard. So I am an alumni of here. I know most of the teachers. Hopefully, most of the teachers know me. I'm seeing in the audience many people I may know or may have been friends with, may have studied with as well. Following that, I did my BSc Zoology in YCI Satara and my Master's in Shivaji University. And I completed my Master's in Bioinformatics in Aberdeen in Scotland. As mentioned already, I don't want to go beyond what has already been mentioned. I've worked in the industry as well as academia, currently working in academia as a bioinformatician. The official post would be research technologist, but I am doing the work of a bioinformatician. So coming on to Omics, as Bakre sir and Chaudhary sir has already mentioned a lot about information about bioinformatics and the scope and all that. I thought today, instead of going too much into detail about bioinformatics, what it is, something which can be, you can always get in a textbook, the general information about it. I want to go across the work that I've done, the practical application of bioinformatics, which would be more interesting maybe to understand how bioinformatics, how Omics is actually applied in research. So essentially, just for the basic knowledge of bioinformatics, what is it? It's known as the science of storing, retrieving, analyzing large amounts of biological data. That is the basic definition of it. It's an interdisciplinary field involving biologists, computer scientists, mathematicians, and also many other fields as well, statisticians, for example. And it forms a major part of modern biology. It forms the heart of modern biology. You cannot think of biology, any sort of field of biology without bioinformatics being part of it now, given that there is a lot of data, a lot of information available all over, and there is needs for analyzing that data, for organizing that data, for putting it in a way that it can be made useful for further research. So what sort of data are we looking at? I mean, as mentioned, as both sirs have mentioned before, there's lots of data in biological data, for example, chemical data, biochemical data, metabolomic data, all this data. We have means of creating the data with the modern technology, with the next generation methods and techniques which are available. So what do we do with that data? Can we use that data to enhance our science, to make it more easier to understand? So yes, that is what omics and what bioinformatics, the field of bioinformatics, is used for majorly. You can see that there are various branches to it. Bioinformatics is an umbrella term, if you like, under which all these different fields come under. So you've got computational biology, transcriptomics, metagenomics, drug development, which is very important nowadays, understanding protein structure and proteomics, genomics, understanding evolutionary studies, how were things before, how have they evolved. So all of this comes under bioinformatics, or omics as it is shortened to. So just go beyond. One of the most important, I guess, applications, I mean, when we talk about practical applications and the various fields of omics which are used, one thing which comes up right now, we are all on our phones and computers listening to a seminar which could have been done in an auditorium sitting next to each other live, but it's not happening. The reason for that is we are have a pandemic on our hands at the moment. Now, this is a very serious pandemic compared to earlier ones, and the severity of it, I think, can be reduced by the scientific knowledge, by knowing, by finding out what the infection is, how we can go about reducing the infection, getting rid of the infection, and essentially omics or bioinformatics or the various fields of bioinformatics are playing a major role in that. For example, I'm just highlighting a couple of papers which I found very interesting in the use of bioinformatics for understanding the COVID. For example, the paper in Nature where they're talking about understanding the knowledge, understanding the information about it, and understanding how it's infecting, where it's moving towards, how can we move it, what cells does it affect, etc., etc. So the information is there, is found from medical information. We can move on to say, how do we understand the virus? So going into not only the infection, but now the nitty-gritty of the virus. What is its structure? What is the sequences? What parts of the virus are vulnerable? Where can we find a cure for this? So all the tools which are there in bioinformatics, they can be used for finding this out. We can look at the proteomic structure, for example. Where does it lie? What sort of receptors does it have, etc. There's so much research available, and the speed at which the research is being done is mainly because of the use of bioinformatics, newer technology to understand all these viruses, and especially this one. So coming back to being a bioinformatician, what sort of skills does one include? I mean, the skills can be numerous depending on what field of bioinformatics you want to go into. What sort of, because bioinformatics, as I said, is an umbrella structure, what sort of skills do you need to accumulate? So I personally have accumulated these skills. I'm just going to list them out here. This is something which you can look at as well. So for example, the ones in green are all basically biology-based skills: molecular biology, cell biology, systems biology, genetics, etc. So these are skills which I have accumulated over my studies, over my experience. Now, as a biologist, computer programming can seem very, very difficult, very, oh, that's not what I can do. But no, actually, programming is very easy because the logical part of it, once you understand it, you can get to it. So what sort of programming skills would you look at? So you look at database management, you look at programming, scripting, you look at web development, you look at cloud management. So these are all tools by which the data which is handled can be stored, can be presented, can be analyzed, etc. So these are the tools which you, from the biology and the computational part, which you get and which you require. And of course, the core bioinformatics skills that you require, the bioinformatics databases which are available online, the bioinformatics tools, which tools would you use for what analysis, knowledge of that? Data science, given that you have so much data, what tools would you use to analyze that data, to interpret that data, to show for people who do not understand anything about the data, a layman person, if I tell them about it, I should be able to explain to them what I'm talking about. So that's where data science comes in. So once you have accumulated all these skills, if you look on this, on the right-hand side, these are this diagram essentially maps out what I have done personally. As I said, bioinformatics is a very varied field, and you can go from A to B in a multitude of ways, funnily enough. So I went down the computational biology way, where I was using computational skills to solve and apply to biological data. So including algorithm development, software development, database construction, etc. So this was the computational part, and now application of it in bioinformatics, where we did sequence analysis, function analysis, structure analysis. So these sort of modules, if you like, are what I specialize in. I look at, I deal with most of these skills which you see on top in this table. So this is where I work. I work in the University of Glasgow, which is currently just in the latest rankings have come. We're 92nd in the world in world university rankings, which is essentially a good place to work in. Also, I work in a laboratory called the Dow Davis Laboratory, and the people you see in front of here, this is Professor Shireen Davis, and this is Professor Julian Dow. These are my principal investigators. These are my bosses, if you like. They run this lab, and it's a lovely lab, lots of people always there from multi-cultures, multi-countries. It's a very fantastic lab to work in, not just from the people point of perspective, but from the perspective of what the lab actually does. So this is where my zoology background comes in. So this lab works in systems biology, which is again an offshoot of that. We work with cells, cell biology. We work with physiology. We work, for example, what you have in front of you is the Malpighian tubule. This is the Malpighian tubule, which is essentially analogous to the human kidney. So this is the kidney of a Drosophila. The Drosophila, or fruit fly, as we know in India, as the I think it's called the banana fly. I'm not sure, but it's a very, very small, minute organism which we study. This now, why do we use insects for as models? Now, as we know, as a zoologist, we can use various organisms as models. Like I have used, when I was doing my master's, I used mice as a model. Essentially, we cannot go and just experiment straight away on humans. So we use ethically, we use insects as models for our studies. Right. So if I can go back, sorry. So human diseases. There are various human diseases which are known, and basically all these, the fly, the worm, the yeast, these are used as models. So these are examples of examples of the diseases and stuff which are found in humans, which are studied as in models. So as you see from the diagram on this side, why insects? Because they constitute most of the living organisms. So this is a representation of the number of organisms there are on the earth right now, of which insects are the largest proportion. So therefore we use insects as models, and Drosophila being the most important one, we use that one. Now, what do we use? We use Drosophila in our lab. We use the Drosophila as a model, but what are we studying? So we're studying different kinds of fields of science. We're looking at basic science, we're looking at insect biology, and we're looking at biomedicine within our lab. So within there, if you see the highlighted parts, the metabolomics, transcriptomics, functional genomics, these are essentially the ones which I'm using at the moment, I'm working on at the moment. But we're also looking at kidney stones, inborn errors, modeling systems biology. So these are different things which our lab is working on at the moment. One thing which I work on, which is the most important question, is what do genes do? So as you can see, this is the Drosophila. This is the pie chart describing the Drosophila genes with all the different various tasks that they do, functions that they do. So my job is to figure out what genes are known, what genes are not known, and how to find them out. So that is my task, finding out what genes do. Now, what we have done in our lab is a couple of resources that are very well known all across the globe. So I've helped in creating many of these resources. So one of the main ones which is the Fly Atlas 2, which is a Drosophila gene expression atlas. This app helps you to find out what genes are present in what genes are present in which tissues, and what the expression is. So this is one app which we created. Another app which we created is for the metabolomic atlas, called as FlyMet. Now this allows you to see the various pathways, the metabolites which are present in Drosophila, and it shows you lots of information on that. So as I said, I was part of that as well. One of my main projects, of course, was NeuroStressPep. where I designed a web app called Diner, a database for insect neuropeptide research. So neuropeptides, I'll come on to neuropeptides in a minute, but this essentially was a resource for understanding what neuropeptides are available, are present in insects, and what sort of so what information can we gain from neuropeptides? So I'll just go on to the next slide. Professor South already mentioned this, but NeuroStressPep is a huge Euro Horizon 2020 grant, which I was working on. We were working with 14 collaborators all across Europe and America. We were generating, what the basic idea was to use the neuropeptide information of insects as a pesticide. So the hormones, we were targeting the hormones, so trying to make it a greener pesticide rather than using chemicals to get rid of pests. We were targeting the insect, the pest's own neuropeptides and the regulatory systems. So this was a huge effort, and I was lucky enough to be part of this project. So what are neuropeptides? Neuropeptides essentially are chemical messengers that circulate in the body and control bodily processes. Neuropeptides are responsible for regulating a variety of physiological functions, such as growth, development, digestion, metabolism, ecdysis behavior, water balance, iron homeostasis, etc. So I do apologize. I don't know why my computer seems to be rushing ahead. Right. So from this project, we had quite a few publications, of which I was, again, lucky to be part of. Most of my work was published for that project. And what I'm going to do right now is I'm going to focus on one of these projects and explain to you about that project. Now, if you give me 1 second, please. I don't know why my presentation wants to run away. Okay, hopefully it will not run away. So this paper, which I'm going to talk about, is called The Evolutionary Trends of Neuropeptide Signaling in Beetles. This was essentially a comparative analysis paper where we took the transcriptomes of 30 different beetles, which were found to be either pests or beneficial insects. And we wanted to find out the neuropeptides. We wanted to identify the neuropeptides in these species and then target them to see what sort of evolutionary significance was present in these. How could we differentiate between pests and non-pests? How can we find out a particular pesticide which will affect only the pest insects and not affect the beneficial insects? So that was the main study. We wanted to look at all these beetles together. So as I said, we looked at 30 different insects. We looked at their transcriptomes and we looked at their genomes as well. So we got all the information from all the different classes, subclasses of these insects. And in the methods section, you'll see that we used quite a few bioinformatics methods for assembling. Now, what does that mean? When we're assembling a transcriptome, essentially the data that we get from the sequence, the insects, is in the transcript data that we get, the mRNA information. Now, this information, when we do it using sequencing methods, we get a lot of raw sequences. These need to be put together to form a big transcriptome, which will give you the information about all the genes that are available in that insect and what genes are expressed, what genes are not expressed. So we need to assemble these. Now, assembly can be done either one where you know what the information is, and you can just align the information that you don't know against it. Now, in these cases, there was no reference sequences. So what we had to do is we had to use techniques whereby we do de novo assembly. Using de novo assembly, we assembled the transcripts of these sequences for their genes. Now, using as I said, all these methodologies which you see on the right side of your screen, FastQC, digital normalization, Trinity, Busco, etc., were used for the assembly. For the neuropeptide prediction, we used these different software, the ones which I've listed there. And finally, for phylogeny, i.e. finding out the evolutionary significance of these neuropeptides, we used tools such as Cluster Omega, CLC Workbench, B10, WebLogo. These may be just words to you, or maybe you have used them. But don't worry about that. Right now, just understand, I'm trying for you to understand what process goes in the whole project of finding out gene information. So this is a little pipeline which I created. So as a bioinformatician, you're expected to create pipelines. Pipelines meaning, now there is no time limit on how much time a process can take. Now, you can't just sit on your desk, wait for the process to finish, and then move on to the next process. So what you need to do is you need to program or script a way that all the processes can be done one after the other with the information from the previous process and go ahead and till you get the final results. Now, this is a pipeline which I created. So I'll just go through the pipeline quickly, where all the different modules of these pipelines was created by me using software which is either already available or creating new modules so that the work would happen in a sequential manner. So essentially, from the samples which we got, the tissues that we got, we moved to transcriptome assembly. We did the assembly. We then did the neuropeptide prediction, as you can see, with the tools which I mentioned earlier. These tools have to be done in a certain way. And so we did all this. We did the sequence alignment. We did determination of signal peptides, peptide cleavage site prediction, and then identification of the precursor sequences and the mature sequences. We then also looked at the receptors of these using different methods, alignment method, non-alignment method, and we got the information for the receptors. Now, finally, we did the multiple sequence alignment. So we looked at conservation between the genes, which we then looked at identification. We looked at evolutionary significance using phylogenetic trees, sequence conservation. What is sequence? Is there any motifs? Are there any patterns there? What sort of domains are there? What sort of transmembranes are there? So we did all these studies because more information is always good. No information isn't. So as much information as you can gather, as much information is there, and you can make better results and better interpretations from that data. So if we look at the results that we got, this is just some of the examples of data. Now, this data is a visual representation of all the information that you have. You need to be able to present it to people to explain what that data did, what that information you have. So, for example, you have here your BOOST score over here. The BOOST score gives you the validity of your data. You have KEGG pathways out here, which means what sort of metabolic pathways, what sort of genes are there which are present, what sort of different metaboloids are present. You can find that information out. How many sequences were found in each database? What are the biological processes, the cellular processes, etc. Gene ontology, gene annotation, functional ontology. So there are many aspects of this which you can study and which are important for the further analysis. The main part of this project, of course, was neuropeptide precursor and mature peptide identification. So finding out the genes which expressed the proteins. We needed to find out what those genes were, where they're present, and how different are they, if at all, between the different insects. This we did by looking at the sequence data. And once we got the sequence, we got nucleotide data first. We translated it into protein data. And then each sequence was checked, was analyzed to see what sort of protein it was coding for. Where are the cleavage sites? So where are the mature peptides? So a lot of information can be gathered just by looking at the sequence of the gene or sequence of the protein. And this is what we did here. We identified the sequences of the neuropeptides. For example, FMF-R-imide, allostatin B, CCHMI. These are examples here. We looked at 50-odd different neuropeptides in 30-odd different insects. So you can imagine the amount of work it was me who was doing it, how much work it was went into this. So from the information that we got for the 30 insects, we created a neuropeptide profile. Now, if I was to give you information in the form of sequences, that would not make sense to a person who wants to quickly find out some information. So not only is bioinformatics analyzing the data, but also you need tools to present the data. You need to make sure that the person, the audience who's looking at your data, who's trying to understand it, should be able to easily understand it. That's the best part of science, is sharing what you find. And that is what we did here. So we created a profile for all the neuropeptides that were found in all the insects. From that information, we looked at conservation. As I said, it is important to look at patterns in these sequences. Are, as you know, ATGC, for example, they're very, when you look at sequences, they seem random, but they are not random. They are very, very much, their patterns are formed between the sequences. So one other task for verification of your sequences is to check your pattern, the patterns which you find, against patterns which are known already. So this can be done using something called as multiple sequence alignment, where you align all the sequences one under the other to see the conservation which is present. So as you can see on the left-hand side, you can see the conserved sequences which are there, the patterns which are formed. You can distinguish the patterns and say, yes, okay, this particular pattern exists in this protein, therefore that peptide must be present. So this is another verification process, as well as finding out the conservation. On the right-hand side, you may see the different sequences from the different insects that we saw, and we can see that all the sequences are lined up under each other, and the conservation is very high. So you can see that is how we can say, yes, those proteins exist, or those genes which code those proteins exist in our data. So next thing we did was we looked at evolutionary trends. This is essentially the last thing which we did, which I will talk about. What does it mean by that? Well, essentially, we want to check for particular peptides or for particular insects, what is the evolutionary significance? Which insects share a similarity in their genes, in the proteins that they code? Which proteins, if for example, beneficials are together or the pest insects are together, it is then ideal for then you can targeting these genes, for targeting these proteins. If they're not together, then we'll have to do further work. So a way of finding out the significance of the validity of the peptides is by looking at the evolutionary significance. How have they differed over the years? Has there been any changes? How similar are they? As you can see in the SNPF on the right-hand side, they are very, very conserved, and they have been consistent throughout the ages. So many of these beetles are many years old, while many beetles are very young. I say young in the age is in millions of years ago sort of a divergence. So I'll come to that. But so we're looking at using tools to identify the evolutionary significance of this. And similarly, we made more profiles. Now, not only did we make profiles on just the evolutionary significance, but also other factors, which I'll come to. So if you look at the evolutionary significance here, the top ones are the very, very old ones. These are, if I can just get the age, 190 million years ago, these were diverged. So these are very, very old, the 201. So you've got very, very old beetles at the top and very, very young ones at the bottom. Is there a difference in the neuropeptides? You can see that the more younger they get, the more neuropeptides are identified in them, while the older ones don't have them. Is that of any significance? So these are questions which we ask when we look at this data. Similarly, based on their diet, can we make any significant conclusion on their diet? So you can see that herbivorous ones are different from predaceous ones, that there are more neuropeptides which can be targeted. Or more importantly, you can specify which neuropeptides you do not want to target if they are herbivorous or if they're predaceous. So there's lots of interpretation which you can make from the data that you have in front of you. And as I said, interpretation is important, not only presentation is important. So you need tools to do that. Finally, I'll look at size. Does size matter? The size of a beetle, of an insect, Is it a pest? If is it beneficial? Does the size matter? So from the size, you can see that the larger insects do not have many neuropeptides compared to the neuropeptides in smaller ones. This is very interesting as well. Does that mean you can target some neuropeptides in the larger ones, which will not, in the smaller ones, which will not affect the larger ones, and vice versa? So there's a lot of assumptions we can make, a lot of interpretations we can make. And essentially the summary from this is, using pipelines, we can assemble, analyze sequence data of more than 30 members of the Coleoptera, the insects. We've identified their neuropeptide precursors. Species are very varied. We looked at different species, suborders and orders of the species. We looked at the evolutionary development and trends of the genes. We looked at conservation. We looked at differentiation of the neuropeptides across the species. And we did this to get a better understanding of their functionality. Now, that's essentially what I wanted to show you. I wanted to go talk to you about the work that I did in a practical sense. So you understand biology, bioinformatics is something which is, it's not a specific field. You come across many different fields, many different opportunities, many different research subjects in it. It does not need to be very targeted. But as you go, you gain a niche field of expertise, which is something which I've done. And the research that you carry out, as you know, is very important. And being a bioinformatician, you play a very, very big role in research nowadays. And as Bakr sir has already mentioned, as Chaudhary sir also mentioned, job opportunities are immense based on your skill set. So if at all you're looking at a career in that, definitely keep your eyes open, keep yourself as much open to different skill sets, etc., to progress. If you have any questions, please feel free to ask. If you want, rather email me. You can get me at that email address which is on the screen. I can, if you have questions, if you have questions about career, particular research, etc., please feel free to ask me. No, not a problem at all. Thank you very much for the opportunity. Thank you.
Very nice, Aniruddha. Now, for some time, we should spare for open discussion, I think.
Yes.
So if somebody wants to discuss with Dr. Aniruddha Pandit, may raise the hands or may comment in chat box.
Please ask your question in the chat box or raise your hand. I will unmute you and you can directly talk your questions. It is a request to all the participants that please raise your hand or give your question in the chat box.
What is current situation, Aniruddha?
In terms of what, sir?
In terms of employments and in terms of routine in COVID, during COVID.
All right. Well, essentially, yeah, many universities, many companies essentially had to shut down, especially in Scotland, England. They did have to shut down. We were given an option if we wanted to work from home or if we wanted to be furloughed. And people took their decisions accordingly, because most, for example, I mean, in terms of laboratory-based work, they can't really go to the laboratory and work in this case. But as a bioinformatician, I had the advantage that I could just pick up my computer, take it home, and work from home.
So experimental work has become difficult now.
It has been, yes, it has become quite difficult. Thankfully, the infection rate out here has reduced.
Or working with animals is also now becoming difficult.
It is, yeah. Yep.
So what is possible solution work? What is possible solution?
For the virus?
For laboratory working.
Oh, no, no. Okay. Well, essentially, our laboratories, what they have done is they do a very, very intense cleaning of the laboratories. Everybody use protective equipment. We are social distancing ourselves
Yes.
in the laboratory. So we're trying to keep as safe as possible, but the work has to continue. So the labs have opened up again, but everyone is
Okay.
taking care of each other.
Is it possible now to working with animals?
It depends on the animals. I'm sure, yes, because if you keep the animals in a sterile environment and a safe environment, then it should be. I mean, for example, the University of Glasgow CVR, the Center for Virology Research, they're actually working on the vaccine. They're part of the team which is working on the vaccine in the University of Glasgow. So they are working with viruses and especially with the COVID virus. So you can imagine how safe and how protected they must be keeping themselves and everybody else.
Yeah. अनम्यूट करा की त्यांना डॉक्टरांना अनम्यूट करा सर. Dr. Shantanu Abhyankar, welcome to the room. Yes, you can talk now.
I've already put my question.
Can any protein
be called the first protein? Can any protein be called first protein?
No, I wouldn't say that. I mean, if you discover the gene, if you discover the protein, then that would be the first. There are more than 30,000 genes, essentially, and not all have been studied yet. So any protein that you do discover, I mean, it's an immense honor to do that. But yes, our laboratory itself, we've been working with aquaporins. We've been working with neuropeptides, for example. We have discovered novel neuropeptides, and if we are able to present our research effectively, then yes, we can name our proteins and call them the first proteins. But it depends.
Okay. There is another question from Dr. Pratima Pawar.
Sure.
She is asking about postdoctoral opportunities in Aniruddha sir's lab.
So essentially, all laboratories out here, all laboratories post their job opportunities online. The best way, I mean, I would give advice to anybody who's looking to do postdoctoral studies, postdoctoral work, employment, is to approach. The best part of working in the UK or US, for example, is approachability. If you approach the principal investigator and give them, show them your CV, tell them your interest, try and gauge what the information is, there are always opportunities.
Okay, now from Dr. Madhuri Patil, which type of free softwares are available for polygenetic analysis?
Phylogenetic, well, basically, the best part of bioinformatics is most software is free. So all you need to do is Google the software, Google what you want, and you will get it. Why is it free? Well, most software is free because it's created by scientists, and out here scientists are considered as very poor people. We don't have money to buy commercial things, so we create our own software. And then we create our own software and we give it to other scientists to use for themselves. So that's why most of it is free.
Okay, thank you.
Oh, that's.
What is career after biomedical science?
I think Abhijit would be better suited for that reply.
Okay.
This is not my field, unfortunately.
Abhijit sir will be joining shortly.
Yeah.
And there are several comments that they have liked your presentation. A very nice presentation, Aniruddha, from Dr. Abhijit Mane. Thank you, sir, from Varsha Kumbhar and Priyanka.
It's very kind. Thank you. I hope I wasn't too fast. I did not want to.
It's okay. It's okay. Very good.
Yeah.
Dr. Bharat Bhalerao, which green method is used as insecticide, but whether it covers your area or not, I don't know.
No, that's fine, because this is essentially was my project.
Okay.
The green insecticides, that is what we're looking at. Many green, there may be green insecticide. A green insecticide is defined as one which does not use harmful chemicals, which will, what happens is, if you go to a field and spray a chemical, you may get insects that are beneficial, who are actually useful for pollination or other, for getting rid of pest insects, may also die because you're just spreading chemical generally. So what we're doing is we're looking to remove that, create targeted insecticide, targeted pesticides, and which are not harmful to the environment. So which will not harm the beneficial insects. They may not harm the crop. They may not harm the soil. So that's what we're in the process of developing.
Thank you very much. If somebody wants to talk with resource person, may raise the question or any point of discussion. Dr. Abhijit Chaudhary is now in our room. I welcome him on behalf of all participants and all my colleagues.
Thank you.
So thank you, Aniruddha.
Okay. Thank you very much. Thank you, everyone. Thank you.
So as Dr. Abhijit was not available due to time lag from California, now he has entered in our room, and I wish a very heartful welcome on behalf of our Janta Shikshan Sanstha's Kisanir Mahavidyalaya Y and on behalf of our organizing committee. And now I hand over the charge to Dr. Sunil Sawant to introduce Abhijit Chaudhary. Thank you very much.
Dr. Abhijit Chaudhary is an associate professor. Dr. Abhijit Chaudhary is an associate professor of radiology at the University of California, Davis, USA. After completing his Bachelor of Engineering in Electronics Engineering from University of Pune in 1999, he joined California State University, Northridge for his MS in Electrical Engineering. There, he not only completed his MS in 2002, but also successfully completed his PhD in Electrical Engineering in 2007. Simultaneously, he completed his one more MS in Applied Mathematics from the University of Southern California, Los Angeles in 2007. Dr. Chaudhary specializes in methods for medical imaging inverse problems, medical image analysis, imaging of musculoskeletal and neurological disease. His Chaudhary Lab is located at the Department of Radiology, UC Davis School of Medicine, and at the California National Primate Research Center, University of California, Davis. This lab aims at developing and validating novel medical imaging techniques with a focus on diseases of the musculoskeletal and neurological systems. His research group utilizes a range of imaging modalities that include MRI, PET, SP, CT, ultrasound, and optical imaging in both preclinical and clinical settings. His areas of interest include medical imaging, image analysis, inverse problems, image acquisition and reconstruction, imaging biomarkers, arthritis, neurodegenerative disease, etc. Several of his research papers have been cited several times by the worldwide research scholars. As a principal investigator, he has so far successfully undertaken the following research activities. First is total body PET-CT for assessing rheumatoid arthritis, disease activity and treatment response for the period 1st of August 2019 to 31st July 2022. His second project is quantifying synaptic density loss in a monkey model of early Alzheimer's disease. This is for the period of 1st August 2019 to 30th April 2021. Third project is imaging biomarkers of early synaptic changes in a preclinical model of Alzheimer's disease for the period 1st of August 2019 to 30th April 2021. And one more project is quasi-rigid image registration for DC MRI. For the period, it was completed long back for the period 1st Jan 2013 to 31st December 2014. Dr. Abhijit Chowdhury is the son of Dr. J.H. Chowdhury, the secretary of our Shikshan Sanstha, Janta Shikshan Sanstha Y. He completed his schooling in Panchgani, a hill station famous for its international standard educational complexes. Now, he has carved a niche for himself in the academic and industrial world by making a solid contribution to research in biomedical science. I invite, on behalf of Janta Shikshan Sanstha's Kisanveer Mahavidyalaya Y, Dr. Abhijit Chowdhury to make his presentation on biomedical science.
Thank you very much, Dr. Sawant. Respected guests, Dr. Jayant Chowdhury, the Secretary of the Janta Shikshan Sanstha, Professor Bhadirao, the Principal of Kisanveer Mahavidyalaya, Dr. Sawant, thank you very much for the kind introduction. Professor Bakre, I really thank you for inviting me to this session. I do see a number of acquaintances. As Dr. Sawant described, I almost grew up in Y College. My dad has been working with Y College for a really long time, and therefore I am acquainted with a number of the folks that are on here, including I see Dr. N.V. Chowdhury as well. So firstly, thank you very much for this opportunity. We know that we are in difficult times, but despite the COVID-19, it's really great that we can connect via this new medium of Zoom calls. And therefore, I've always maintained that the world is becoming a more and more global place. And I think that's what we are really valuing during this time. The second thing that I would like to bring up is, of course, the opportunity of speaking today, which is Teachers' Day. And September 5th is a day that all of us honor. So to all the professors on this call, all the teachers, all the individuals who actually teach other people, this is a real honor for all of us today to be here and to celebrate really the legacy of education and the legacy of Dr. Sarvapalli Radhakrishnan, who has really set the agenda and the vision for education in India. And many of us, whether we are in India or whether we are in foreign countries, are really descendants, legacies of Dr. Sarvapalli Radhakrishnan. So I think that he would be really proud to see us all here. I'm going to share my screen now to start my presentation. Can everybody see my screen?
Yes, sir. Yes,
Perfect.
sir. Please continue.
Perfect. All right. So the topic that I'm going to talk about today is about the role of animal science in biomedical research. And as Dr. Sawant introduced, I am with the Department of Radiology. I am also a director of a major center that focuses on animal imaging. across multiple species, including non-human primates. And I've just shown some examples of the types of imaging methods that get used across different species. So in my presentation today, I'll start briefly with my own background. And you might be wondering after that introduction as to why am I qualified to talk about imaging about animal science, because my background is as an engineer, and I trained in engineering and mathematics and electrical engineering predominantly. So how would I know a thing or two about animal science? So I'll actually describe my own background in that area. Then I'll talk about animal science and how it's revolutionizing everything that we do today in biomedical research. And it is really a great opportunity for me to share this seminar with Mr. Anirudh Pandit, as a person who actually looks at genomic level aspects per se, to kind of my own expertise, which is looking at kind of the entire body and the animal level aspects. I think we complement each other's expertise, and therefore I'll be talking about where I see the major contributions in medical research and drug discovery of how we are using animal science methods. I also will then generalize my description to exposure and risks in humans, and how animal models are providing us with a novel way of looking at aspects. And then finally, I'll end with some remarks about climate change and how animal models are actually helping, or animal science in general is helping us navigate through this complex topic. Finally, I'll actually talk about some careers in biomedical research, and really conclude with remarks that careers in animal science are highly important. Don't let anybody tell you that there are some careers are more important than others, and hopefully I'll make the case for it. I also want to state that animal science plays a critical role in making biomedical breakthroughs today, and that will be the broader theme of everything that I will be talking about today. All right, so to start out with, our family comes from Kenzar. Many of you might be familiar. I grew up partly in Wai for earlier years before my parents moved to Panchgini. That's a photograph of both of them. My education was in Sanjeevan Vidyalaya, Pasgani, and subsequently got my 10th and 10th through 12th, 11th through 12th and 12th in SP College, Pune. Following that, we also entered engineering college, and then I was a student of electrical or electronics engineering as part of the University of Pune. I came to the US in the year 2000, first finishing my master's degree in California State University, Northridge in electrical engineering, and then subsequently moved to the University of Southern California, where I did my master's degree in applied mathematics and a PhD in electrical engineering. So if you look at my profile, mostly it represents that of an engineer that has gone on to now becoming a professor in the field of radiology. However, let me actually tell you where my connections with animal science began. This was my PhD thesis paper that actually is a very well-known, well-documented paper in the literature, where for the first time we actually started to study animal models of cancers, in this case brain cancer. So what you see in the figure, can everybody see my mouse?
Yeah.
Perfect. So what you see in the figure is essentially an animal model of cancer, and you see some signal that's coming out from the brain. And so this is an animal model of glioblastoma multiforme, or brain cancer. And so my association with this area in terms of research started where I started to design systems that would help in imaging of animal models of disease. Subsequently, after I finished my PhD and I joined the University of California, Davis, I now direct a major center that focuses on imaging of animals. And the reason why we image animals is because we are trying to assess disease and follow disease, just like we do in humans. You get CT scans and MRI scans and so on and so forth. In a similar manner, our university has a large center where we have pretty much all the scanners for different imaging modalities that you get in humans. You actually have them for small animals as well. And this is a snapshot of one of individual from our lab, Charles Smith, showing the different suite of scanners that we have. The center is called the Center for Molecular and Genomic Imaging, and I have been directing the center for the last 4 years. Also is my affiliation with the California National Primate Research Center. Now, again, I am one of the core scientists at this center, and also direct the animal imaging program at the center. What this center is that it allows researchers the resources to perform major experiments in non-human primates. As I will go on to explain, we have small animal models such as rats and mice and so on and so forth, and they are good for certain conditions. But there are other conditions where non-human primate models actually are really, really important. And I actually oversee a portion of my own research program that works on non-human primates. And then finally, you really can't get away from animal science at our university because the University of California at Davis actually has one of the longest history of having the world's top vet school, as well as one of the world's top agriculture program. And therefore, we have a number of collaborations across many different disciplines. And therefore, I feel qualified to give this talk about animal science in biomedical research. All right, so let's get started with the content. Now, the first thing that I would like to mention really is the CV of a lifesaver. And if you think about animals that actually have saved lives, basically, you should first think about the laboratory mouse. This slide really describes the tremendous amount of contributions that we have had in the field of sciences by using mice. And so up here in terms of its CV, you have education, and it actually went through all the universities that have any research program use the lab mouse. It has been a topic of so many different Nobel Prizes. Remember that a number of Nobel Prizes have been given for fundamental research, either resource person Mr. Pandit will tell you, Drosophila, insects, and so on and so forth have been a major area where Nobel Prizes have been awarded. At the same time, a lot of Nobel Prizes in medicine, chemistry, and others have also been given to individuals who worked with mice, and they have contributed to things like TB. How does TB transmit? That's 1905 Nobel Prize, to most recently looking at stem cells and reprogramming of stem cells, and this is Shinya Yamanaka's Nobel Prize most recently. So again, huge number of contributions of animal science over here. Lab mouse is involved in 75% of all research that takes place in the world. And because they have 98% genes that are comparable with humans, so we actually are able to manipulate the genome of the lab mouse to make it suitable for our studies. Of course, I have also listed the research areas actually, and of course they cover pretty much any and every disease that we are able to study. So really, the fundamental components associated with the concept of the lab mouse are really important. Of course, as I'm speaking about this, I have to be reminded that animal science was fundamental to the development of concepts of evolution. In fact, if you look back to the history of how we started to study evolution, it's evolved with studying beaks of birds actually in Galapagos, and again, animal science kind of being at the forefront of what we today understand actually as evolution of the environment as well as us as humans. So um, I'm going to now give a few examples of how animal models are used quite commonly. And this is an animal model of what I would say induced cancer. What individuals have determined over a lot of experimentation is that you can create tumors or human tumors in animals. One possible way is to take cells from tumors in humans, inject them into animals, and these cells can be grown inside the animals and resemble these lesions like tumors in humans. Then you can use these animal models to study these tumors. Uh, how do they start from a single cell or multiple cells? How can they grow? What is the rate of growth? And also how we can treat them. And so, again, the models, animal models, are such critical tools for being able to do that. My own experience in this area, of course, and is shown here. Number of cancers today are studied in preclinical models. I've got an example of glioblastoma multiforme. This is an MRI scan showing a lesion in the brain of this animal, very much like what you would see in children, in humans. And these tumors are deadly tumors, and they are problematic. Therefore, studying how glioblastoma multiforme is initiated, what are the triggers, and what are the scientific, what exactly is needed for the tumor to progress, become large, uh, how does the cancer spread, and what can we do to treat it, are all some of the things that we can discuss. The second example in the blue and red over here is kidney cancer. And this is studying the process of what is called glutamine metabolism. How do tumor cells get energy, and how can they grow? And this is in the context of kidney cancer. Again, major cancers in the world currently we are able to study and develop treatments for these cancers. Now, on one hand is induced tumors, which basically means that these animal models don't get tumors naturally. We actually induce them either by injecting of human cells or actually manipulating the human or manipulating the genome of these animals. The other example that we have is in our nature are of spontaneous cancers. And spontaneous cancers are cancers in animals that actually occur like they do in humans. In humans, of course, cancer typically is not induced unless, of course, you are working next to a radiation detector or some such, and the radiation causes the cancer to start, or you are part of one of these physical events per se where cancer is induced. Mostly the cancers that we know are spontaneously occurring, which basically means that sometimes there is a known cause and other times there is no known cause. Well, in the animal kingdom, spontaneous cancers happen in other species. What is shown over here on top is a dog with osteosarcoma in a manner that actually is very, very similar to osteosarcoma in children. At the same time, of course, oral cavity cancers basically in wildlife are actually very, very common. This is an example. Of course, it's not very clear where the cancer is, but it's in the oral cavity of this animal. And this is a classic example of squamous cell carcinoma in an animal model. So there are not just induced cancers in animal models, but spontaneous cancers very much like what we see in human beings. And here is an example actually of an image of a dog with osteosarcoma. That's that hot spot that you see in the limb over there, in a manner that's actually very, very similar to sarcomas in human children and even older adults in terms of sometimes soft tissue sarcoma. So again, we have a wide range of animal species that get cancers in a manner that's quite similar to humans. The other thing that, of course, is really, really important is to consider companion animals. So, of course, you have animals in the wild, and then you have companion animals, the animals who live with human beings, are pets, dogs, cats, etc. Now, these animals get exposed to the same toxins that human beings get exposed to. If the owner of a dog is smoking, the dog actually inhales the smoke. Therefore, we are seeing across the world an increase in the number of cancers, spontaneous cancers in companion animals. And these companion animals, in some ways, are models of what is happening to us as a human species. So it's really crucial to know that animal models are already providing information about cancers that affect humans in a manner that affects humans and their environment. So really important contributions in the field of cancer. Now, here is an example where we can now look at treatment. And I was actually saying first that you can use animal models to determine treatment. This is actually an example. On top, you actually see in this case, a mouse model, and it has a breast cancer tumor in its flank. So you've taken breast cancer cells from humans, implanted them in this animal model. And what you're seeing is what happens when you treat this tumor. And so if you don't treat the tumor, that's the plot underneath where we are seeing time on the x-axis and volume on the y-axis of the tumor. If you don't treat, the tumor keeps on growing, for example, and that's the blue line. And that's expected. That's what happens in humans, is that if you don't treat cancers, the tumors will grow. What happens after you treat the cancer? So here is a treatment. In this case, it was rapamycin. You see that rapamycin is quite effective in breast cancer. So you actually see the tumor kind of reduce in terms of size. Now, what happens actually when the tumor has reduced in size and you stop the treatment? Again, we always, when we think about cancer treatments, we actually have a chemotherapeutic treatment or chemotherapy for a certain duration, and then we stop the chemotherapy. So what happens after you stop the chemotherapy? And what this graph shows is that the tumor size grows again. So the tumor comes back. Now, let's say at this point, you hit it back with the same treatment for a second time. What we see is that decrease in tumor size that happened because of first treatment does not happen because of the second treatment phase. So the tumor has now developed resistance to this treatment. And this is exactly what happens in humans, is that you expose individuals, give them chemotherapy, and you have a reduction of tumors. And over a period of time, they will develop resistance to that chemotherapy, and the tumors will figure out how to grow. And we can replicate this exact process in humans in animal models. Therefore, it provides us a tremendous opportunity for being able to study these cancers. So to summarize actually how we are using animal models in cancer prevention and treatments, we are looking at how can we prevent cancer, for example, and treat them. We are looking at how do you optimize therapy. You have thousands, maybe even millions of different options in terms of therapeutic targets. What do you choose? What is the most effective? We can actually use them in animal models and demonstrate whether they are really effective or not. And they give a pretty good clue about whether they are effective or not in humans. And of course, we can look at toxicity, because a lot of cancer treatment is associated with toxicity. And we are able to now evaluate in animals, given their similar genomic nature, or genetic nature per se with humans, we can now examine the toxicity profiles as well. And at least get early data to know whether a certain treatment is going to be extremely toxic or not as toxic. So really crucial contributions to the field of cancer by animal science. And of course, biomarkers. Some of the biomarkers that we derive from animals are the same ones that we derive from humans. And biomarkers range from anything between your lab tests, by blood tests, for example, to more complex tests that happen in the context of cancers per se, through blood, through saliva, through other fluids of the body. All right, so that's cancer. I hopefully have convinced you that animal models are really important in cancer. The second area that animal models contribute to significantly is neurodegeneration. What I'm showing here is a condition called Alzheimer's disease. So it's part and parcel of a large, broad range of conditions that are crucial from the point of view of neurodegeneration, and it's a condition that affects the aging population. So as you age, your brains essentially start to lose tissue, And at some point in time, it is possible that you might develop Alzheimer's disease and you can see brain atrophy. Now, of course, this is what happens in humans. And a lot of times, by the time we actually know that an individual has Alzheimer's disease, it is too late. So we don't really know what treatments we should really use to treat Alzheimer's disease, because we are already looking at a person who's very late in the disease state. So animal models are again becoming really important. And this is actually an animal model of Alzheimer's disease, where uh, there is a knock-in of two major genes that are observed in heredity-based Alzheimer's disease. So about 10% of Alzheimer's disease is driven by heredity, and this animal model, and I will, and as Mr. Pandit mentioned, per se, pretty much in the similar manner, in animal models, we can manipulate the genome, and in this case, we have made this rat to express these two genes at a much higher rate, and these are responsible for Alzheimer's. And so what you see is a healthy brain, and what you see in kind of the brighter signal is, everything is intact in a healthy brain. And of course, when you have Alzheimer's disease, you actually have a loss of a lot of this signal in the brain over a period of time. And we can now use these animal models to study treatments. We can use these animal models to study how diseases, start, and so on and so forth in the context of Alzheimer's disease. Now, someone might argue that, well, rats are not very smart. Humans are really, really smart. And how can we really replicate, these complex functions, executive functions and motor functions that humans have? And therefore, we move to non-human primates. Non-human primates are smart animals. They essentially kind of belong to the evolutionary tree of human beings, and they actually are capable of a lot of complex functions that human beings are capable of. Therefore, animal models such as that in the non-human primate offer a novel way for us to examine again exactly how does Alzheimer's disease start. Older monkeys actually exhibit standard signs of Alzheimer's disease, pretty much like humans. Not all of them do, a fraction of them do, pretty much like humans. So a lot of interesting concepts can be studied in these non-human primate models that we are just not able to evaluate otherwise. So again, major contributions to Alzheimer's disease and your neurodegeneration research is currently being performed at many different institutions that use animal models, and animal models are contributing to these disease conditions. And then finally, or kind of maybe the third example that I would like to give, and it's kind of been on everybody's mind right now, has been infectious diseases. Several years ago, we had a problem with Zika. And remember that your microencephaly essentially was passed on from the parents to the child, and produced children who had certain features that showed that brain development was not complete. Now, the exact same animal models could be brought in to in this context in a non-human primate model of Zika. So we actually now can study exactly in the non-human primate model how Zika was transmitted. In fact, we got to know about the routes of transmission of this infectious disease through animal models. We didn't really know, because a lot of times what happens in infectious diseases is you don't really know the right routes of transmission. How is the virus transmitted, for example? And we actually have to use animal models to understand better what that is. And of course, has been on everybody's mind has been COVID-19, SARS-CoV-2. What is the problem? And so what I'm showing here is essentially a CT scan through livers of humans, where you are seeing these, what are called ground glass lesions. And these are actually primary pathologies that you see in the lungs of individuals, where you see this mucus formation, and that leads to breathing challenges and so on and so forth. In this context, of course, we are using animal models again, because apparently you can look at COVID-19. In fact, when the initial studies came out of China about COVID-19, they showed that non-human primates actually have COVID-19. They can get COVID-19 and they can transmit the virus very much like human beings can. Therefore, right away, a number of institutions across the world that have the ability of studying these in non-human primates started to study what are the routes of transmission. What happens basically if the parent is infected? For example, a pregnant animal is infected. What does it have? What happens to its offspring? What happens to its child, basically, or kind of the animal, young animal that is given that is born when the parent actually has one of these conditions? So these are types of questions that people across the world are starting to ask using animal models. Of course, uh, there has been tremendous number, and I think you might be reading in the broader press, there has been tremendous number of studies of therapies, vaccine studies, antiviral studies have all been initiated in non-human primates and other animal models to really understand the routes of transmission. Also, as we know, a lot of infectious diseases such as some virus, a range of viruses affect our, farming animals, for example, quite commonly, in the same nature that they affect human beings. And therefore, as on one hand, we as a community have tried to solve the problems of associated with hunger and so on and so forth across the world, and farming and agriculture is so key to the Indian setup per se. The farm animals and so on and so forth, and infectious diseases in the context of these farm animals are crucial things that we have the opportunity of studying, and also studying how these are these particular infectious diseases are transmitted. The other component, of course, I have to mention is immunity, and a lot of what we know about immunity against infectious diseases also comes from basic knowledge about the what we know about animals. Because, and I think as Mr. Pandit also pointed out, the biodiversity that we see in animals is basically many orders of magnitude higher than than the diversity that we see in humans. Therefore, it just provides us a way of better understanding nature and a number of these conditions then that would actually be important for human beings to study. So major contributions again to infectious diseases are continuing through animal science. And then I'm going to get into some complex factors here basically as well, which of course affect us. When you sit in your car and you're actually going to Pune from Hawaii, you are sitting in the sitting in your car and you actually see a lot of smoke around us. This is a common occurrence basically pretty much anywhere you go in the developed world per se, that we get exposed to a lot of smoke and different toxins per se. Question is actually, what do these toxins do to us? And what do they do short term? What do they do long term? And some of our collaborators actually at our university said, let's ask this question. Let's actually take a preclinical model, a rat model, and let's put them into tunnels for about 6 months, and let's see what happens to these animals. And what they were observing here is that actually after exposure to traffic smoke for a really long, for some duration of time, we actually start to see stuff in the brain. We start to see, executive function challenges basically in the brain. And so again, really pertinent to the developing and developed world is this whole concept that we are subjecting ourselves to pollution, and really animal models are providing us a way of understanding that. Now, of course, this is an animal model, but in general animal science, because for example, a lot of times people are carrying their pets with them in their vehicles as well, and so they are also getting exposed to smoke. At the same time, of course, there are these exposures are not only limited to, let's say, traffic related exposures, but being in India and Maharashtra, one of the big challenges that we have had is actually pesticide poisoning. And they have number of casualties, and of course casualties is just a very quick way of quick and dirty way of looking at the impact of pesticide poisoning, it's a lot worse than what has been stated on this slide. And this is actually just within a year. Because, of course, we are not talking about all the other comorbidities, other conditions that get, such as sickness and illness and so on and so forth that come because of pesticide poisoning. So that is a huge problem. And actually the question is, how can we study pesticide poisoning and how it's actually poisoning human beings? Of course, we all know that pesticides have a major role in a farming community such as ours. But we have to understand better what are the influences that these pesticides have on the humans that are spraying them, humans that actually are consuming some of these pesticides indirectly as products, to say. And so that's another area, for example, where what is being shown over here is that a component of pesticides actually, and if you look at how a number of pesticides that we have right now were originally started out as nerve agents per se. And these nerve agents many years ago, about 50 years ago or so on and so forth, have in limited amount because they cause seizures and so on and so forth, in lower concentrations, are being used as pesticides. And therefore, they do have neurologic effects. And what is shown over here in an animal model of neurotoxicity is in this perfectly healthy rat. If you expose this rat to pesticides, basically on a kind of on a continuing basis, you start to see brain atrophy, and you start to see major changes in the brain that would affect the normal functioning per se. So again, really current important topics that we are learning from animal science. And again, I'm giving an example of an animal model here, but of course you can imagine the farm animals that we are using also being exposed to these. And so these animal models, of course, are giving us ideas about how do you prevent toxicities that are induced by these types of environmental factors, and of course treatment. Now, my talk would not be complete if I don't talk about climate change. Across the world, we are facing this issue. Different types of, this year, basically, Australia had, just within the kind of the last year or so, basically, Australia has had these wildfires that destroyed a lot of species from this planet. There have been major fires, as you might have heard, in the United States. We don't know what's coming next. Nature across the world has changed significantly. We have everything from air pollution to water temperatures changing significantly, is what actually this map shows. Is there has been a rapid change in temperature. Now, this is going to affect us whether we like it or not. It is already affecting us in many ways, but it is going to affect us. The specific one I would like to talk about is domoic acid. And that's why I've actually shown the coast of the United States, the west coast of the United States, where you see this rapid heating. What has happened in this area is there are certain algae that require warm temperatures typically to bloom. And of course, when the temperatures were cold previously, we did not really see this algal bloom. But now we see this algal bloom. These algae produce a neurotoxin called domoic acid. And this gets into shellfish and fish that actually live in these areas and feed from these algae and planktons. From the fish, they can get into seabirds and cause tremendous amount of damage. They can cause damage to marine animals. And of course, via shellfish and so on and so forth, they actually can can indirectly have an impact on humans as well. So this is just one example of what climate change actually is doing. And again, animal models and animal science is so critical to understanding how the changing environment is affecting the kind of livelihood of humans across the world, and how it's affecting the biodiversity. Specifically, the coast of California has had a number of challenges. And in fact, this is an unfortunate picture of these sea lions across the California coast that have their population has been reducing because of domoic acid poisoning. Another major area of or something that animal science continues to contribute to, because the toxicities actually that you see in animals basically are just a bellwether, a precursor of what you would see in humans at a later point in time. So really important aspects. And really, I don't think that we have to think of ourselves as isolated. I mean, I think humans learn how to dominate species, of course, but really we are part of an ecosystem. And this ecosystem consists of everything from the microbes to kind of large species, and everything is dependent on each other in one way or the other. And unfortunately, the changes that we are seeing in the environment not only I think it's short-sighted to say that, oh, they affect only kind of one species or another species. In some ways, they affect everybody in some way. So I think anything that we can do to kind of understand the effects of climate and climate change on the animal kingdom is really a way of understanding what it will be in humans pretty soon, and therefore it provides us a motivation for actually doing better in terms of our outlook on climate change and policies that we have to put in place, and environmental policies and other types of policies that we have to put in place for addressing these. And of course, animal science, again, being at the forefront of being able to provide us this knowledge. So lastly, I'll actually talk a little bit about the careers that might be suitable for individuals in animal science. Of course, there is the careers in research, and careers in research are not just limited to academics. A lot of times, I think there is a notion that these lab-based research labs are actually in educational institutions. That's really not the case. Major industries across the world run extensive number of labs that are relying on animals and animal models as ways of research. That includes all major pharmaceutical companies in the world, the Pfizers and the Mercks and the Genentechs and so on and so forth. All of them have large animal colonies where they study animal science, and that therefore you require experts in animal science to be able to design these experiments correctly and to really provide us the input that we need to understand how treatments work, how diseases actually progress, and so on and so forth. So really, many number of jobs actually in the field of research. I do not need to emphasize more. Mr. Pandit is also on the line. Computer programmers and statisticians. It is an unfortunate aspect that, of course, India has produced quite a lot of computer programmers, but on the other hand, we have not really kept up with fields such as statistics and epidemiology. And I think there is an opportunity there, because and it's kind of becoming obvious right now when we are in this health crisis of COVID-19, that we don't really have a solid group of people who are being trained in epidemiology of diseases. And it's so important to know what the prevalence and incidence of diseases are. How do you actually probe the prevalence? What statistical models are important? And it's exactly the same thing that you need in animal science as well. Is that what are the if you are studying an infectious disease basically in animals, what's the routes of transmission, replication, etc., etc. You need people who understand the statistical component, and so really you see a merger of fields. And I don't think that individuals who are trained in zoology should be shy about getting into maybe a master's program in statistics, for example, because that really changes your perspective. And of course, conventionally programmers, we used to, I think the conventional notion in India is programmers actually write software for companies per se, but really in the biological sciences, there's tremendous development in terms of software. I'm sure Mr. Pandit has talked about it. Technical writing is really important actually, because what we have to do together as individuals who have an education per se today and have the ability of propagating our thoughts, having thoughts per se about things, is being able to write them in a manner that can be understood by people. And so we need good individuals who can write well, who can write accurately and communicate accurately. Really, really important. Teachers and professors, I can't emphasize that more. Today is Teachers' Day. Really, really important role in promoting animal science. Of course, we need veterinarians and animal care technicians, whether it is actually taking care of our companion animals, whether it is actually looking at animal husbandry and so on and so forth. Again, a big agricultural community that we have in India, and of course our dependence on agriculture. Then of course there is maintenance, design of animal housing facilities, lab facilities, and so on and so forth. So this is just a subset. One this morning actually, as I was getting ready for my presentation, something that just jumped to my mind, and I did cover it in the presentation, is food science. I mean, if you think about what has happened to food per se, 50 years ago, starvation across many countries was actually a common occurrence and a common cause of death. And I think to some extent, the globalization actually has helped, and our supply chains are better at providing food per se. But the consumption of food by human beings and by animals, basically, who are in the same environment, is an important area. And then food science per se is such an important area that it needs to be emphasized. And of course, there are multiple careers in that specific area. And so in summary, I hope I have convinced you that there are promising opportunities ahead. Really, the sky is the limit in terms of what individuals in animal science can do. I think, compared to many of the other professions, I find that individuals who are trained in animal science actually are more versatile and more knowledgeable about a lot of aspects than narrow fields that some other disciplines might be taking. I'd also actually challenge everybody to think big and broad. A lot of times it's actually, when you think about animal science and you are in a course, basically, you are just focusing on what is being provided by that course. If you are studying Drosophila, you focus on Drosophila. If you're studying, let's say, a specific infectious disease, you just focus on that infectious disease. But let's think big. Let's try to think about how can what you are learning impact us in terms of human health, human welfare, and so on and so forth. And how can we improve everything around us per se. And I talked about some large aspects: environmental factors, climate change. These are important aspects that we have to address, or you have to address, basically, as the younger generation. And really, I mean, I like to kind of leave you with some thoughts about kind of a skill set that I think is really crucial. It has been very crucial for my own career development, and I hope I don't sound too sectarian in here. But reading is really important, because read newspapers, understand how economics and how communities and how healthcare and how animal science are all connected. How are we part of an ecosystem? Important to communicate, learning to communicate, verbal, written communication. Because again, I am sitting in my house in Sacramento, California. Mr. Pandit is sitting in his house in Glasgow, in the UK. You are actually sitting in your own homes, in your own spaces, in different areas. We are communicating via the common means. So of course, globalization is already here, and therefore being able to communicate is really crucial. I would also challenge everybody to think. I think a lot of times when we are performing a certain course or we are performing a certain job per se, we get limited to what we see in front of us. We don't think broader, and I think that has led to some challenges. And we don't put aside time to think. In fact, one of the things that I do, and I have had a number of students that I have trained till now, is actually I have them put time aside to think. Because once you're busy, you don't have time to sit back and think. And it's really important to think about how you can contribute and how the knowledge that you are earning can contribute. Of course, plan and implement is really crucial. And of course, that is something that that I would challenge everybody to do. So that's really all I had in terms of my presentation. I'm going to leave this last slide up, and well, again, thank you everybody for giving me the opportunity of speaking here and expressing our thoughts. And of course, going back to what I started with, your animal science is really important, and careers in animal science are really important. And I think I hopefully have convinced you that animal science has been crucial in providing major breakthroughs in multiple areas. And I'm hoping that these breakthroughs will continue, and you will be a major part of or participate in these breakthroughs in a minor or major way. So, all right, thank you very much.
Thank you, Abhijit. You have explained very nicely. And now we are heading towards discussion. So I request Professor S.P. Kamay sir to take Aniruddha and Abhijit together for discussion. And our chief guest, Dr. J.S. Chaudhary sir and Professor Narendra Chaudhary sir in one room. There are some questions from participants.
Sure. Let me maybe start.
Palimane asked that, sir, I have a question that chemotherapy treatment can cure the cancer fully. Can it reoccur?
Yeah, so that's a really good question. Yes, in theory, every cancer can reoccur. And part of the problem is that, on one hand, our bodies actually have learned to adapt and change constantly. That's exactly how we have survived for so many centuries. In that process, what has happened is that our body at some point in time will develop resistance to any type of treatment. Question is, when will that happen? Let's say if you are a 60-year-old individual and your body takes about 40 years to develop resistance, then I guess mostly you're okay. If it actually develops resistance in 2 or 3 years, then that's really not okay. And currently, we do not really understand very well who will develop such resistance and who will not, and at what rate. So, of course, there is a lot of challenge, but yes, it is possible that recurrence will happen. A lot of cancer treatment focuses on short-term gains per se, and that's the unfortunate part. But uh, but yes, there is no doubt that recurrence happens.
Thank you. Sir, as you said, COVID can be transferred to animals. So I wanted to know, are there universities or laboratory researching on this to avoid more harmfulness?
Absolutely, absolutely. COVID can be transferred from humans to animals. It can actually be transmitted from animals back to humans as well. This was actually demonstrated earlier. Um, there have been across universities, there have been high levels of precautions being exhibited by individuals who work with animals. The standard protection equipment or PPE basically that you would use for limiting transmission, such as masks and face shields and gloves and so on and so forth, are something that actually people who are working in the animal sciences are also using to avoid that. Now, it is possible that a number of species actually already have developed resistance to SARS-CoV-2. And because it's not that this virus is really new per se. However, transmission can still happen. And yes, there are, again, to answer your question, absolutely there are multiple ways in which you can limit transmission. And it seems that PPE is probably one of the most effective ways for limiting that transmission.
Okay. COVID can transfer from animal to animal. He already has discussed the point. What does technical writers mean exactly? What is technical writers? One
That's a
of the
really good
careers in
question.
animal science.
Absolutely. So that's a really good question. So if you go to any of the places and you procure a brochure, for example, let's say a brochure for a pesticide, who do you think wrote that brochure? Um, and so technical writers actually play roles in multiple areas. Number one, they play a role in securing funding for companies or academic research labs to do the type of work that they want to do. Of course, except for huge companies per se, a number of smaller companies rely on venture capital for being able to do their operations, and reports have to be written. So technical writers are really important as far as kind of writing those reports. Technical writers are also important from the point of view of communicating with the public. You have to know how to communicate complex science, per se, to in a layman's terms, to so that the public can understand why it's so important. One of the nicest things about a number of scientists that we have seen during this COVID-19 process is that they can communicate. And in fact, the scientists who have been quite successful in getting public policy going are the ones who can communicate versus the ones actually who cannot communicate very well. And at the same time, the writing components are really important because written reports are what we rely on extensively. So on one hand, you have to be technically correct, because remember that's not a marketing report where you can fake things to say. So you have to be technically correct. At the same time, you have to be comprehensive and be easy to understand. So you have to really be a kind of master of all these different skills. And so that's what I meant by technical writers in general, who perform this crucial job of communication with venture capitalists, with our public, with other scientists, for example, and they're the ones who write the important white papers, brochures, etc., that are available, are made available to the public for being able to be adopted as public policy and so on. So
Yeah, no, one thing is we are experiencing here in India that many NGOs are putting pressure on zoologists and other life sciences studies that they should not use animals or they should not kill the animals. So what is situation over there in outside India?
see, I mean, I think everything actually is subjected to what we call humane endpoints per se. And so with every study, we have to get approvals basically for that study through either a local or central board per se. Beyond that, we have to justify why we are using animals, and
Definitely.
that justification is really, really important. Part of the justification, for example, would be in the context of cancer. I think nobody bats an eyelid when you're actually using animal models of cancer, because what are you going to do? Are you going to actually induce cancer in human beings to be able to study cancer? I mean, what is the option really? And so I mean, I kind of fall in that category where I believe that pretty much like anything that we do, yes, there are going to be pros and cons.
Yeah.
But the question is, we have to manage risk. We have to manage basically the cost and benefit. And I think in a number of cases, if there is justification for using the animal models, then I think that justification in terms of human health is important. And so yes, of course, we have to use humane endpoints for these studies. We have to have criteria of if you're inducing injury, basically, what are we going to do to minimize the uh, the challenges that the animal has? If we are actually going to perform euthanasia, basically, what is the procedure for euthanasia such that it is humane? And of course, these all are actually covered within the limits of what we typically have in animal models. So I don't think it's correct to pressurize zoologists actually in that sense, because I mean, I think I hopefully showed you that lots of advances of basically
Yeah.
lots of advances have happened
Ethics
because
of-
basically we have had animal science. So,
Yeah, ethics of animal use in that we should follow.
and
To what
absolutely, and
extent?
I think it needs an oversight as well. So because you can't rely on individuals to actually be, but there needs to be
Yeah.
training, which is actually what the universities here insist that be done. There needs to be review of the protocols, and there needs to be auditing of the protocols just to make sure that the appropriate means are followed. So I think all of us are in agreement that we have to be ethical and careful about the use of animals, and therefore that
Definitely.
would be a strategy.
To what extent tissue culture laboratories can help us in this concern?
Um, so
Anna सांगा.
the
Sir.
problem actually is to some extent what happens is that uh, in tissue culture you can get replications of certain disease conditions, but on a systemic level, on a whole body level, the same component is extremely complex. We have had
Yeah.
multiple examples of drugs, for example, that work perfectly with tissue cultures per se, but when they were moved into preclinical or clinical trials, actually completely failed. Um,
Okay.
and one of these examples actually with a company that we were working on, they actually had a drug for treating one type of cancer. They got initially good data from tissue culture, and they decided to actually rapidly move that drug through into they got approvals to basically go into early stage clinical trials, and they found that it was basically not efficacious in humans, and so they had to basically stop the Food and Drug Administration had to terminate the trials. When they went back, they observed that this particular the mode of delivery of this drug was such that the drug got stuck in the kidneys and never cleared out of the kidneys. And so, unless you are actually looking at the human as a complex system, or kind of the body as a complex system, you just don't know what the biodistribution is of these drugs. You can't really, just treat cells. And so it's very selective. I think that's a good starting point for tissue cultures, but, it doesn't really help when you actually have to move to a human clinical study based on, those data.
Thank you very much. Mr.
I
Dr.
think
Mane.
Abhijit Mane, sir.
Dr. Abhijit Mane, please continue.
Dr. Abhijit, I have one question for you. Right now, you said that there are certain reports of COVID-19 infecting the kidneys. But are there such reports that COVID-19 is infecting the RBCs? Because my certain readings found out that that RBCs are also being infected with the COVID-19 virus.
Yeah, and that's a good question. So some of the kidney effects are actually there have been some reports of kidney effects. I think the bigger effects are on the lungs because you do see mucus formation in the lungs. I would say that it's an evolving area right now in general. I mean, I think lots of reports are coming out at this time, and I have not kept up with the exact literature about RBCs, but I do know that there have been certain integrins, for example, and white cells that actually have been implicated in fibrosis that you see in these tissues, and so that might be kind of a more fundamental, at a more fundamental level. It might be related to some of the components in blood. but I don't know exactly about the question, about how to answer your question. It's still a moving target, though. Unfortunately, every day I see new information about and we are still learning.
Thank you, Abhijit Mane, for your contribution. Mr. Aniruddha, you want to talk something?
Yeah, go. I mean, I almost am talking single-handedly. Go ahead, Aniruddha.
No, I'm actually listening to you as well. It's always good to learn. The wet lab is not part of my repertoire, if you like. I'm more of a
Yeah.
I'm just a computer guy.
Yeah.
So no, thank you for answering those questions. It's good to hear. Good to hear the answers from an expert of that.
Yes, you want to conclude something about
No,
Abhijit's
as Abhijit said, with regards to the question regards to the COVID, of course, as he said, it's still unknown. The main target is, of course, the lungs, the respiratory system, how it's affecting it, and what is the after effects of that is also of concern. It may or may not affect the other organs of the body, but there is also definitive knowledge that it is affecting children, infants, in some way as well. So as Abhijit rightly said, it's still need a lot to know about it. Right now they're focusing on a vaccine for it, but and everybody's rushing. I think there's a question here saying, when will the medicine be available for corona? I'm not sure the vaccines can be done overnight or over a couple of weeks. There is a long process for it. Medicine per se, there's no medicine for corona. It will have to be a vaccine, and it needs to go through a lot of trials, clinical trials, lot of testing, even before they think of giving it to the public. So yeah, it's unknown at the moment.
And I totally agree with that. The challenge, of course, is that in the process of desperation, we don't want to make mistakes, and I think that's the challenge that all of us are faced with, because I think everybody wants to move fast, and and of course, economies need to open. We need to take care of the large, still considerable amount of debt that actually is happening. Of course, there are direct impacts on the lungs and the respiratory system. There are impacts on other organs that we are just starting to understand. However, the bigger question is we have to do good quality clinical trials that involve the standard randomization and phase 1, phase 2, phase 3. We have to go through that process. We really can't rush it. Because ultimately a vaccine, when available, will basically be injected into millions of people across the world, or billions of people across the world. So if we get something wrong, and 10 years later we actually have a problem, then it'll be billions of people who will be affected by that problem. So I think caution needs to be exhibited about when exactly. Now, of course, there are multiple attempts, as Mr. Pandit said. There are Astra, basically, and then Oxford, of course, have been working together. So has Moderna and several other companies in the US. There was a report actually of a vaccine from Russia. I think that came up in the last 24 hours. There has been some development in China. There have, of course, there are different strains of coronavirus as well that have been implicated. And so whether these vaccines will actually be catch all, or will be an evolving component, such like the flu vaccine, for example. Every year it's a little bit different. Will it be one shot, or will it be multiple vaccines that you will have to take? These are all questions that we actually are still in the process of finding out.
Just add to that, please do not just think about miracle cures, and read properly into the news. Do not just believe what you see and hear on WhatsApp. So many things came up before, ginger, hot water and stuff like that, which is absolute rubbish. make yourself aware about things before even taking any sort of medicine or whatever, miracle cures or whatever. Yeah.
And, yeah. I
That can be
can't
most
emphasize
harmful.
that more. Yeah, I can't emphasize that more. Actually, just within the I mean, whether it's WhatsApp or whether it's any of these, it's so important nowadays to fact check. And really, if you get a message, basically just type it into Google and see if actually there is any confirmation about it. There are confirmed sources per se, and I think in this pandemic has made it worse because people are just following instructions unless they come from a legitimate source. ICMR in India, the Food and Drug Administration, or the CD Center for Disease Control or some such. It is really not good guidance, and unfortunately, there are a number of health reporters who are also kind of jumping on the bandwagon of putting things out. So critically thinking through things per se is really crucial, and really not believing the first thing that you see, but looking for validation is really important, especially in these WhatsApp and so on and so forth messages. I mean, it's unbelievable actually what type of things that actually get popularity.
Absolutely. No. Somebody just asked a question, which I think Dr. Abhijit has just answered. Are laboratories across the world collaborating with each other? They should. Good science is always collaboration. Pharma companies may, however, just to answer that question, pharma companies may be looking to make a bit of a profit as well. So take it with a pinch of salt. Collaboration is good. Collaboration is definitely happening. As Dr. Abhijit just mentioned, many labs across the globe have been working together or are sharing information with regards to coming up with their vaccine. There will be, of course, people or companies or countries who would not want to share the initial information, but hopefully it will be available for everyone once it does come out eventually.
Yeah, and to some extent, actually, at least in the US and as far as Moderna is concerned, and I think it's true with the Oxford vaccine as well, is that academic institutions, or in the US, the National Institutes of Health, which is a major publicly funded organization, is actually involved in the process of validation per se. But it's absolutely essential. I mean, if you look at most recent papers in journals such as the Lancet, for example, you would see that there's a lot of international collaboration that's going on. It's amazing how people are coming together during the pandemic to have meetings and have things going. So yes, absolutely, and of course sharing of information. I think that's something that that we saw right away when when the initial information about this coronavirus came out, and actually there was some amount of information that was not made public in the beginning. That led to a lot of problems basically that we are facing now. Had some of that information even in December of last year been made public, then perhaps we would have done better in terms of containment. So I think it's almost become necessary now to collaborate. I also see a question about the Russian claims about the vaccine. Now, I can't really confirm those or kind of deny those. But what I have seen, though, is that the initial studies have been done in 38 subjects, if I'm not wrong. That's what the BBC report has. And so that's actually a small number. And a lot of times, when you think about clinical trials, you actually have to think about them systematically. The phase 1 is a safety and efficacy is the end. Safety is the endpoint. So you give the drug, and is it safe? Is the only question that you can answer. You can't really answer a question about efficacy because you don't really evaluate that. Phase 2 is the efficacy component, and you have to evaluate efficacy. And then the phase 3 basically, phase 2 and phase 3 basically look at kind of the effectiveness, routes of administration, etc., etc. So if you are seeing an early stage study that is predominantly needed for safety, and you're seeing that there are conclusions that are being drawn about the efficacy, then that is a problem. So I think we have to look carefully, and I know that the Russian group has published in Lancet. I have not had a chance to review that paper, but it seems like a small study with number of samples equal to 38. So, so I think I mean, as a scientist per se, I don't really have any issues with anybody coming up with an effective vaccine, really. I mean, as long as it's effective, whether it's Russia or whether it's China or whether it's the US or whether it's, England. I mean, I don't really have any problem with that. But what I have a problem with is if their vaccine is not well tested and well validated, then I think I have a problem with that. So I think that's really what I'm always looking for is the devil in the details in many ways, on exactly what was the protocol, how was the patient population selected, were these people who were very severe in terms of COVID. Now that does that actually I mean in the context of vaccines you almost need individuals who basically are asymptomatic, basically, right? That's kind of how we give vaccines per se, is that you don't really have the disease basically when you're testing a vaccine. But is the immune response basically appropriate? What type of antibodies are being created? I mean, these are all questions, of course, that need to be addressed. And can the antibody? What is the lifetime of the antibodies? Are they staying in the body for long? How much time is immunity being given for? And so on and so forth. So these are questions, and therefore, I mean, I think it's going to be a stepwise phase to really understand what the effectiveness of these vaccines will be.
Okay, thank you. Now the only last comment from Dr. Vaidya. Whether the laboratories across the world are collaborating with each other for vaccines?
Yeah, I think Aniruddha answered that question is absolutely yes. I think there is arrangements of sharing samples. There is arrangements for sharing data. There is arrangements for sharing, methods for analysis. Whether it's the stuff, for example, like what Aniruddha is involved in, or more so kind of stuff that we are involved in. There are big arrangements, and in some ways actually always research has worked that way. Every year I'm sure Aniruddha does it and we do it, is that we go to these conferences where we present our data in a public forum and really the data get peer reviewed, they get improved basically, and they get put into manuscripts and published. So absolutely the efforts are always scientific efforts are mostly collaborative, because even if you think about somebody going to a conference and looking at your data and saying, oh, I don't agree, or I agree with something, that actually in some ways collaborating with somebody on a certain project and making science better. So absolutely, I think most of the people in science actually will tell you that yes, we are more than happy to share whatever we are understanding and collecting.
I'll just quickly add to that yes, yeah. I mean, apart from that, apart from the actual scientific methods, the techniques which are there, resources, bioinformatics resources, databases, software, etc. All of the many commercial companies actually made freely available for the study of COVID to scientists. I mean, some stuff would cost thousands of pounds, dollars, etc. was made free, so that this information, I mean, these tools are actually very, very specialized. And to be given free access for this purpose was actually very good. So that also happened. I mean, everybody in their good nature, everybody does want the benefit of, for getting stuff, for getting, like, for example, the pandemic under control, or, get it studied. So they will definitely go over and make things available, make things free, so that, and shared. And that is essentially good science. That is what science should be. And as Dr. Abhijit actually said, sharing of your information, sharing always leads to better stuff, rather than if you keep it to yourself, you're not helping others. I mean, everybody cannot be an expert in every field, but if two experts come together, they can make more advancements in that.
Definitely. Thank you for this. Now we are heading towards the conclusion, and I request Professor Dr. S.R. Sawant for concluding remarks, and then Organizing Secretary Dr. H.D. Kanse will offer vote of thanks. Thank you very much.
Yes, I would like to congratulate both Mr. Aniruddha Pandit and Dr. Abhijit Chaudhary for making their presentations. We came to webinar, and we had one expert from bioinformatics and another from animal science, and we had a very wonderful evening where both experts shared their research and different insights uh, across different disciplines. So once again, I congratulate both the experts, and now I would like to conclude this session of discussion and request Dr. Kansey to propose a vote of thanks. Dr. Kansey.
Respected dignitaries and participants, I, Dr. Hanmantrao Kansey, Organizing Secretary of this international webinar on biomedical science and bioinformatics, would like to propose vote of thanks on behalf of our Janta Shikshan Sanstha, Kisanvir Mahavidyalaya, Department of Zoology and IQAC. I express my sincere thanks to the resource persons, Mr. Aniruddha Pandit, bioinformatician and web developer, University of Glasgow, United Kingdom, and Abhijit Chaudhary, Associate Professor, Radiology, University of California, Davis, USA, for sparing their valuable time and making insightful presentation in our international webinar. I also wish to express my deep sense of gratitude to Dr. J.S. Chaudhary, Secretary of our Janta Shikshan Sanstha YE, for gracing the inaugural ceremony and sharing with us his vision. We are extremely grateful to our principal, Dr. E.B. Bhalera, for meticulous planning and an eye for details in the organization of this event and chairing the inaugural session. I am grateful to Dr. Sunil Sawant, member of Academic Council, Shivaji University, Kolhapur, for introducing all our guests and resource persons. I thank Mr. Ravindra Bakre, convener of this international webinar. My sincere thanks are to Mr. S.P. Kamble, coordinator of IQAC, for hosting this international webinar. I have no words to express outdate of gratitude to the management, vice principals, all my colleagues, all the participants from different countries, universities, colleges, the administrative staff for making this webinar a huge success. I convey thanks to all distinguished delegates, participants, invitees, and well-wishers for their active participation in this webinar. Thank you very much.
Okay, fine evening to everybody. And as a convener, I would like to thank all the participants and the guests and resource persons. And now I request to our Zoom coordinator, in charge host of this room, to conclude the session and to make the data off for this room. Thank you very much.
Okay. Thank you, sir. Now the meeting is ends here. Okay.
Thank you, everybody.
Thank you.
Thank you again, everybody.
Thank
Thank
you.
you.
Yes,
Thank you.
see you again. Aniruddha and Abhijit.
Thank you.